So we have been using high-density grids to study stroke and also spinal cord injury. And although we have a long-standing interest in motor unit behavior, to begin with, we have been focusing mainly on spatial properties of the EMG distribution across impaired muscles. Many of you know we changed our name. We were called Rehabilitation Institute of Chicago, but for strange and mysterious reasons, we are now called Shirley Ryan Ability Lab. And my colleagues involved in these studies include Ghulam Razul, Nina Suresh, who is here, Babak Afsharapur, who actually trained here at Lisin, and Surav Chandra. We begin by asking some rather elemental questions about the way in which activity is distributed across impaired muscles. And we know, perhaps not surprisingly, we know relatively little. It's not surprising because we've not had access to mapping tools to be able to show us the way in which different parts of the muscle behave. But indeed, we know very little about this right now. So the first question we're interested in is, does the spatial pattern of muscle activity activation change after a hemispheric stroke? The second question was whether this disruption of the spatial pattern was somehow linked to changes in the intrinsic mechanical properties of muscles after hemispheric stroke. We are now learning in regard to that second question that muscles begin to deteriorate quite quickly after a stroke, and over a period of time they may lose muscle fibers, muscles become much stiffer, and there are a lot of changes in architecture of the muscle that are becoming clear on ultrasound and other measures, which we don't really understand. We don't know where these changes come from, but ultimately they have a substantial adverse effect on the quality of performance of that muscle and ultimately on the quality of life and motor performance of the person. So the question here was whether there were electrical analogs, whether we could see using high-density EMG recording changes that would reflect some of the intrinsic changes in architecture of the muscle. And then the third question, more pertinent to spinal cord injury but also to stroke, is the availability of the high-density grids gives us the opportunity to map innervation zones. So another question we can ask is whether these innervation zones are changed after a stroke or spinal cord injury. So, yep. This is a setup of a 264-channel grid sitting over the biceps muscle of a stroke survivor. One strip is over the long head of biceps, and the other one is a short head. And people are being given instructions to do isometric voluntary contractions, as shown in the lower trace. Next slide. And we draw these, we call them heat maps. These are the maps, like Deborah showed, for the pain, how the muscle activity changes as a function of contraction level in the muscles. And on the top left is one head of biceps, the top right another, and then on the bottom we are showing a progressive increase in background isometric force measured in relation to maximum voluntary torque, and this is done in this instance from an intact subject. And you see this sort of like concentric maps with the highest activity level in the center and a systematic distribution of muscle activity through the muscle. To begin with, I don't really understand why the maps look that way. I think it's because the thickness of the muscle, of course, is largest towards the center. Therefore, the largest muscle mass lies there. But on the other hand, we have estimates that tell us that the depth that you can record from the surface is relatively small. So how is it that the thickness of the muscle is still being reflected somehow in the distribution of muscle activity? This is something that could be tested experimentally, of course, and with modeling and simulation. But as we sit right now, I don't really know why the maps look the way they do. But this gives us a template, nonetheless, for how to expect a normal biceps brachii to behave, and conversely a biceps in someone who's had a neurologic disorder. Next slide. So the key pictures here are on the top. I should say, just to clarify, these are normalized to the max in each case. We're not seeing progressive increases in EMG. This is mainly to look at the differences in shape distribution of the regions of activity. On the top, we can see from both long and short heads that the activity distribution in a paretic muscle of a stroke survivor has changed substantially. The map has been spread out much more, and we are seeing systematic high levels of activity over much of the surface, as compared with the contralateral side, which is shown below. Next slide. And so we can begin to ask certain questions about these distribution of the maps. One is how consistent these maps are. And these are not easy to see, the slides, but the key point is that the map consistency, perhaps predictably, is somewhat greater in people with the consistency is greater when you compare normal controls against stroke survivors, and it's also a more consistent map distribution from the contralateral side of a hemiparetic stroke survivor as compared with the impaired side. Next slide. And this makes the same point. The blue is a reflection of normal muscle, and the upper colors are just looking at the distribution of the center of mass of clustering of these contour shapes between impaired and contralateral muscles. Next slide. But then, how does this tell us about the questions that we were seeking to answer? And specifically, one question number 2 on my first list was, does the pattern of spatial distribution somehow reflect changes in the architecture of the muscle? Are we seeing the effects of accumulation of connective tissue, of muscle fiber death, or other changes in architecture? And one way that we looked at that question was to look at pixel-to-pixel variation. If there is a different kind of tissue present in the muscle, for example, connective tissue or other matrix, we might expect, that between different pixels of the grid, we would see some seeing higher concentration of muscle than the adjacent recording electrodes. And that turned out to be true. Here we're comparing the consistency of the pixel-to-pixel magnitude between contralateral, therefore unimpaired and impaired sides of stroke survivors, and the black is the stroke, and the white is the impaired side of the stroke survivor, and the white is the unimpaired side. And the greatest similarity, this is a measure of similarity index, was always true for the white as compared with the black side. So we were seeing a lot more pixel-to-pixel variability in the impaired muscles of the stroke survivor than in the contralateral. That doesn't directly tell us whether there is some other tissue present that's maybe changing the spatial distribution of the muscle, of the electrical activity in the muscle, but it's consistent with that idea. Next slide. So the third question we looked at then was this mapping of innervation zones. This was done mostly by my colleague Babak, and it was primarily in spinal cord injury, although we have been starting to do this in stroke. And again, we are mapping the innervation zones using the high-density grids. Next slide. And it's hard to see. What we were looking for was the characteristic feature, which is visible on differential recordings where we have spreading of motor unit action potentials away from some a central point, giving us these sort of V-shaped distributions for the motor unit activity. And you can use these features of the single differential on the grid to estimate where and how many innervation zones are present within the muscle. Next slide. And what we've seen, surprisingly, is that in people with spinal cord injury, there was often a remapping, as shown on the right, of the innervation zones of the muscle. This is coming from a spinal cord injured person in which both sides were impaired, but it looks like on the left biceps brachii, there may have been damage either to motor neurons or to ventral roots, which caused the muscle to re-innervate, caused the motor neurons and motor axons to re-innervate parts of the biceps. So we developed these novel ring-shaped innervation zones, which probably tell us in hindsight that there had been damage to either ventral horn or to ventral roots on that side. Next slide. On top of this, and we expect this, we also see high levels of spontaneous EMG activity in both stroke and in spinal cord injury, some of it which may reflect damage to motor neurons. A lot of it may be a manifestation of spasticity. Next slide. So, just to conclude, what does this mapping using the high-density grids of muscle after these neurologic injuries tell us, or at least begin to tell us? The first thing is, does the spatial pattern of muscle activity within a muscle change after hemispheric stroke? Seemingly yes. We don't quite know why. It could be because the stroke causes people to use different patterns of motor unit within a muscle, which may cause the map to change. Or alternatively, it may be because some of the motor units are damaged, so we are looking at changes in architecture that result from that damage. The second question is this the disruption of the spatial pattern linked to changes in the muscle intrinsic properties after hemispheric stroke? Probably yes, but we have to correlate the changes in the spatial maps using EMG with changes that we see on ultrasound and other measures to be really confident that we're on the right track. And then finally, and perhaps more predictably, we see quite frequently changes in innervation zones after after a neurologic injury, especially after spinal cord injury, but sometimes also stroke. And probably this is a reflection of damage either to ventral horn or potentially to the ventral roots. Thank you.